Hello everyone, welcome to my YouTube channel. Today we'll be talking about the complement system. For some time now we've been looking at the immune system for a while. We've talked about the inflammation and we are still under the innate immune system. Today we'll be talking about the complement system. And before I continue, I would like you to take 3 seconds of your time to hit on the subscribe button to subscribe to this channel. I want you to also like and then comment. Alright, so let's begin. So the complement system, as I said earlier on, is an example of the innate immune system. You know, the innate immune system is not specific. The innate immune system is what? It's not specific. So when we are talking about the complement system, we can classify them into 3 categories. So we have the classical pathway, we have the alternative pathway, and then we have the lectin pathway. Today, basically, we are going to focus on the classical pathway for now, but we are going to talk about the proteins that are involved in the complement system. Okay, so now the main function of the complement system is to clear pathogen through inflammation, opsonization, and then cell lysis. So as we move along, we'll get to know how the complement system clears pathogen through inflammation, opsonization, and cell lysis. Now, the complement system consists of what we call complement proteins, and these complement proteins are in our plasma. They are circulating throughout our bloodstream day in and day out. So these are the complement protein that combines themselves to form cascades that ultimately leads to the clearing of what? Of pathogens. We are going to take a look at examples of these proteins that are found in the bloodstream that contribute to the complement system. Okay. Okay, so this is just a sketch or a representation of the proteins that are involved in the complement system. So normally we denote them with C. So we have C1, C2, C3, C4, C5, C6, C7, C8, C9. So these are the complexes. So we have all the proteins that are involved in the complement system. So we have C1 complex. So the C1 complex consists of isotypes like the C1R, C1Q, and C1S. Then we have the C4. So the C4 can also cleave to form the C4A and the C4B. Then we have C2. The C2 too can also cleave to form C2A and then C2B. Then we have C5. C5 too can also cleave to C5A and C5B. Then we have the convertases. So for the convertase, we have C3 convertase. So C3 convertase is actually what cleaves C3 to C3A and then C3B. Then we have the C5 convertase too. So the C3 convertase, for instance, consists of C2A and C4B that has been combined. And then the C5 convertase consists of the C2A, C3B, and C4B that has been combined. In other words, C3 convertase that has been combined with C3B. C3 convertase that has been combined with C3B to form the C5 convertase. So the C5 convertase will be responsible for cleaving C5 into C5A and C5B. So this is basically the logic behind these complement proteins and then how they work. Then we also have the mannose binding lectin. The mannose binding lectin. This mannose binding lectin is a carbohydrate and is mostly used in the lectin pathway. The lectin pathway. Then we have our membrane attack complex. This is also known as what? The MAC, the membrane attack complex. So the main purpose or the main reason for the complement system is to ultimately result in the membrane attack complex that is responsible for causing lysis to cell or causing cell lysis. So what it does is that this membrane attack complex pierces through the cells and then allows water to enter into the cell. The cell becomes turgid and then it undergoes what? Lysis. So that is the complement proteins for you. So you have to take notice that there is the convertases. So the convertases are responsible for the cleaving of the complement proteins to its subunits. So C5 convertase cleaves C5 into what? C5A and C5B. C3 convertase cleaves C3 into C3A and then C3B. Okay. Now, when it comes to C4 and then C2, it is C1s that is responsible for the cleavage. So C1s is the same protein that is responsible for cleaving C4 into C4A and then C4B, and it's the same C1s that is responsible for cleaving C2 into C2A and then what? C2B. So basically this is all the logic we are going to need to understand the complement system. So first of all, you have to know that they are complement proteins, and these complement proteins do have subtypes, and these subtypes are formed through the convertases. So the convertases is responsible for converting the main complement protein to its subtypes, and in some cases like the C4 and the C2, it is the C1s that converts them to their subunit. That is in terms of C4, C4A, C4B, and in terms of C2, C2A, and C2B. Okay, so this is basically the logic behind the complement proteins. Now the mark consists of C5B, C6, C7, C8, and C9. So all of these ones combine to form what we call the membrane attack complex, or the MAC. So C5B, C6, C7, C8, as we can see over here. Yes. So these proteins combine to form what? The membrane attack complex. And the membrane attack complex is what is going to cause what? The cell lysis. Okay. Now with this understanding, I believe we can proceed to the classical pathway. So this is the classical pathway. So it's a very simple process. There's nothing to worry about its complexity. It's a very simple process. So this is what happened. So when what you have to know is that this complement system are especially the classical pathway, they are antibody mediated. So for instance, in the classical pathway, you can realize that the complement protein, the first complement protein, binds to the FC portion of the antibody. So it could be IgM or IgG, depending on the structure. Okay, we know that the IgM is a pentamer and then the IgG is a monomer. So in this particular case, we can say that this one we are illustrating is the IgG. So let's just consider this to be the IgG. So this is what happened. The first complement protein, that is the C1, binds to the FC portion of the IgG. And you realize that the IgG has already bind to the pathogen. So here becomes our pathogen. Already binds to our pathogen. So the first complement protein C1 binds to the antibody that has already bind to the pathogen. Okay. So this is what happened next. So what happens is that, remember that the C1 is a C1 complex. The C1 is a C1 complex. And I already said that the complement system, they are circulating in the bloodstream. So what happens is that the C1 complex, the C1s, this one, in the C1 complex cleaves C4 into C4A and C4B. So this is what we have here. So you can see from this particular place that C4 has been converted to what? C4A and C4B by the C1s. So the C1s cleaves C4 into what? C4A and then C4B. So now the C4B, this C4B will continue binding to the pathogen. So we realize that we have the C4B over here. Okay, so the C4A, for instance, might dissipate from the bloodstream, and some of them too might also involve in other inflammatory activities, but mostly they dissipate from the bloodstream, the C4A. So what is really important here is the C4B. So the C4B binds, continue the binding. So the C4B binds to the pathogen, and then the binding continues. Now, what also happens is that there is also going to be a cleavage of C2. So there's also going to be another cleavage of C2. So C2, then C2 will also cleave towards C2A, and then C2B. And what is responsible for that cleavage? This same C1 S. This same C1S protein that is responsible for the cleavage of what? C2. So all you have to notice is that, or take notice of, is that the C1S is responsible for cleaving C4 and then C2. C1S cleaves C4 to C4A and C4B. C1S cleaves C2 into what? C2A and C2B. Okay. Now, in the case of the C2, this is what happened. The C2A, this one is the one that is going to bind to the what? To the C4B. It's going to what? Bind to the what? To the C4B. So you can see from this diagram that C2A has bind to what? Has bind to the C4B. Okay. Now, after the binding, this C2A and C4B combines to form what we call the C3 convertase. Remember what we said about the C3 convertase? So C3 convertase is actually C2A and C4B. It's actually C3 convertase. It's actually C2A and C4B. So C2A and C4B forms what we call what? The C3 convertase, as we discussed earlier on. Okay. Now, so this C3 convertase, what is going to be the function? It's going to convert C3 into C3A and C3B. Now, C3B will continue the binding. So C3B will continue to bind to the C3A convertase. In other terms, C3B will bind to C2A and C4B. So you realize that C3B has bind to C2A and C4B. And these three combines to form what we call the C5 convertase. These three combine to form what we call the C5 convertase. And what is the function of the C5 convertase? The C5 convertase is also responsible for converting what? C5 to C5A and C5 what? B. Now, C5B will continue to bind with the other complement proteins like the C6, C7, C8, and then C9. And they will together form what we call the membrane attack complex, or what we call what? The MAC. The membrane attack complex, or the MAC. And this MAC will initiate what we call cell lysis. So it punches through the cell or the pathogen, and then it causes the pathogen to burst or undergo lysis. So basically, this is what the classical pathway is about. Let's go over again. So what happens in the classical pathway? So first of all, the antibody-antigen or IgM binds to the pathogen. The first complement protein, that is the C1, which is a complex in the sense that it consists of C1R, C1 kill, and then C1S, binds to the FC portion. So this C1 complex binds to the FC portion, specifically the C1 kill binds towards the FC portion. And then what happens next? The C1S is responsible for cleaving other complement proteins, especially the C4 and then the C2. So what happens is that it cleaves C4 into what? C4A and then C4B, and it cleaves C2 into what? Into C2A and then C2 what? B. Good. So now, after cleaving C4 into C4A and C4B, C4B will continue the binding. Then what happens next? The C2 will also cleave to C2A and then C2B, and C2A will also continue on the binding. Now C2A and C4B will form what we call the C3 convertase. Convertase means it will convert C3 into C3A and C3B. So C3 convertase will convert C3 into C3A and C3B. So now we have our C3A and C3B. What happens next? Now C3B will also continue on the binding, and it will bind to the already C3 convertase that has formed here. So what happens is that when this C3 binds to this thing already, when it binds to this one, it also forms what we call the C5 convertase. The C5 convertase will also be responsible for breaking or converting C5 into C5A and C5B. And then C5B will bind to C6, C7, C8, C9 to form the membrane attack complex. And after the membrane attack complex is formed, it pierces through the what? The cell of the pathogen, and then initiates what we call lysis. So this lysis will cause the cell to what? The cell to burst. Now what you have to take notice is that some of the complement proteins that do not involve in the binding, like the C3A for instance, can also act as opsonizers. The C3B and the C3A, some of them can also act as inflammatory or opsonizing or opsonizers. So that is basically what the classical pathway is about. It's a very simple process. All you have to know is the convertases, the complement proteins that are responsible for converting other complement proteins, and how all this mechanism is what is initiated. It's a very simple process. When you take your time to digest this particular lecture, you'll be able to understand how this process comes about. Thank you very much for joining this particular session. I would like you to hit on the subscribe button. Please, it's very, very important. Kindly hit on the subscribe button to subscribe to this channel. I wish you good luck. See ya.